Section 11-4 is on meiosis. And if you'll remember, right before we finished with first semester, we talked about mitosis, and now we're going to talk about meiosis. Genes are located on chromosomes in the cell nucleus. This is a review, and each organism must inherit a single copy of every gene from both of its parents, which means you get half from your mom and half from your dad. Each gamete, and if you remember, a gamete is an egg or a sperm, contains just one set of genes. So what happens is the egg, which has the label of N, and N is haploid, combines with the sperm, which is also N, which is haploid. You end up with a zygote after fertilization, which is 2N, and that is diploid. Now, when we think about chromosomes, half of the total number of chromosomes comes from dad, and the other half of the total number of chromosomes comes from mom. And when we talk about this, paternal is dad, maternal is mom. These two sets are called homologous, which means dad matches exactly to the chromosome that came from mom. So then these are called a homologous pair of chromosomes. A cell which contains both sets of homologous chromosomes is called diploid. And what I did here is I made sure both, the important things are in red. The term is diploid, and diploid means two sets or 2N. So in humans, the diploid number is 46, or 2N equals 46. So we have 46 individual chromosomes, or 23 pairs. The gametes of sexually reproducing organisms, now remember the gametes are egg and sperm, contain a single set of chromosomes that are haploid. Haploid means one set or N. So in humans, the haploid number is 23, which is half of 46. And humans have a total of 46 chromosomes, or 23 pairs. Meiosis is the reduction by division of the number of chromosomes by half in the sex cells. And meiosis only refers to these sex cells, which is the egg or the sperm. So meiosis only applies to the sex cells. Mitosis, on the other hand, is for the body cells. So if we have these coming from mom and these coming from dad, the child cannot have 4 chromosomes. That would be too many, too much. So mom and dads have to divide. So meiosis reduces the genetic content so that the child has the same number that the mom and dad had originally. There are 2 stages to meiosis. There is meiosis 1, which is called the reduction division. And you'll notice all of these little phases down here are similar to what you learned in mitosis, but this has a Roman numeral 1 after it. All of these have Roman numeral 1s. And you'll also notice that they all say diploid. Because here's the key. Before meiosis 1, each chromosome is replicated. So meiosis 1 is similar to mitosis, where we start with our original number of chromosomes, they copy each other, and if we go all the way down to telophase, we have 2 cells that have the same number of chromosomes as our original parent. But the gametes can't have this many chromosomes. There are too many for the gametes. So we have to divide one more time. But I'm going to go through each of the phases of meiosis 1 just to remind you, in case you forgot from mitosis, what's going on in each of the phases. Prophase 1, and once again you'll notice there's a Roman numeral after that. Early prophase, the homologs pair up and crossing over occurs. Now, crossing over is going to occur because these chromosomes are smashed together in this inside of the nucleus. They're so compact that they might be on top of each other and some switch places. Late prophase, chromosome condense. Condense means to squish. The spindle forms and the nuclear envelope fragments. And what the word fragment means is, as you can see, it's starting to break down. Crossing over is an exchange of alleles between homologous chromosomes and produces a new combination of alleles. Now what you're going to notice here is this is a homologous pair of chromosomes. A tetrad is another term for these homologous pairs. Each of these is a chromatid. And these 2 that belong to one chromosome are sister chromatids. And the centromere is what connects the sister chromatids together. Now, what happens is because they're so compact within the nucleus, sometimes, and they are sitting next to each other, they just happen to lay on top of one another. And so what happens is then the chromosomes actually, some of the traits will flip-flop from one chromosome to another chromosome. And that's called crossing over. And that happens, once again, in prophase 1. Metaphase 2, metaphase is the same. The pairs are lining up in the middle or along the equator of the cell. Anaphase, they are starting to separate and move to the opposite poles. The sister chromatids remain attached at their centromeres. And remember, the centromeres are going to be those points on the chromosomes. Telophase 1, the nuclear envelope starts to come back together, the spindle disappears, and cytokinesis divides the cell into 2. But remember, we now have to take these chromosomes and divide them one more time. Hence, we have meiosis 2. So meiosis 2 is the equational division, which means we're taking the cells, the 2 new cells from meiosis 1, and we are going to split them one more time. Here's the key right here. Neither cell goes through replication before meiosis 2. Replication only takes place prior to meiosis 1. you'll notice that there is a Roman numeral behind each of the phases in meiosis 2, and we have now gone to haploid. So we've gone from 2N to N. And then the end result is going to be, instead of 2 cells, we're going to have 4, and they're non-identical haploid daughter cells. So here we go with prophase 2. In each of the 2 cells now, the nuclear envelope is going to break down and a new spindle is going to form. Metaphase 2, in each of the 2 cells, the chromosomes are going to line up in the middle or along the equator. In anaphase 2, the sister chromatids separate and move to the poles. So you will notice now that we are no longer paired. There are now individual sister chromatids. They are no longer in pairs. Telophase 2, the nuclear envelope comes back together, the chromosomes decondense, the spindle disappears, and cytokinesis divides the cell into 2. So the end result of meiosis is we end up with gametes, which are egg and sperm. For haploid cells, 1 copy of each chromosome. You'll notice that there's just 1 in the cell, 1 allele for each gene, and many different combinations of alleles for different genes along the chromosome. Because you can see here that part of this has crossed over and the chromosomes have moved into different cells.